The humoral immune response is one arm of acquired immunity. The production of antibodies and their association in antigen-antibody complexes can effectively clear an infection from the fluid of the body. However, many pathogens, such as viruses and certain bacterial species, have the ability to enter inside cells. Antibodies are bigger in size and can't enter the cell. So, the body's defense against pathogen cells and other non-self cells is controlled by cell-mediated immune response. In this topic, we will discuss the role for the T cell in recognizing and eliminating infected cells and non-self cells, as well as their part in priming the pump for the humoral immune response. Effector T cells generated in response to antigen are responsible for cell-mediated immunity. Protein receptors found on different populations of T cells define their role in cellular immunity. B cell receptors directly recognize epitopes on an antigen. However, the interaction between antigenic determinants and T cells depends on set of surface receptors capable of recognizing a multitude of antigenic fragments. T cell receptors are capable of such recognition. There are two major subpopulations of effector T cells, and their receptors determine how the cells will function in cellular immunity. Cytotoxic T cells have T cell receptors and co-receptor proteins called CD8 on their cell surface. This combination allows the CTLs to recognize and eliminate non-self antigens such as virus-infected cells and tumor cells. The other group of T cells, the so-called helper T cells, has TCR and co-receptor called CD4. These T helper cells are divided into two effector classes, one that helps eliminate bacteria from infected antigen presenting cells and the other that helps B cell produce antibody in the humoral acquired response. The combination of T cell receptors and co-receptors allows T cells to recognize and bind to another set of glycoprotein molecules called the major histocompatibility complex, MHC. MHC protein are embedded in the membranes of all cells of the body. At least 20 different genes encode MHC protein and at least 50 different variants of those genes exist. Thus, the variety of MHC proteins existing in the human population is enormous. That is, MHC is also called human leukocyte antigen, and the chance of two people having the same MHC protein is incredibly small. A notable exception is in identical twins. The MHC protein define the uniqueness of the individual and play a role in the immune response. There are two important classes of MHC proteins, and it is to these that the CD8 and CD4 T cells binds. MHC class 2 proteins are found primarily on the surface of B cells, macrophages and dendritic cells. These cells are called antigen presenting cell, APC, because the MHC2 molecules fold to form a pocket in which small antigen fragments can bind. Thus, APCs present antigen fragments to CD4 T cells, which will bind to them with their T cell receptors and CD4 receptor complex. MHC class 1 molecules are found on all nucleated cells of the body. Again, the MHC1 molecules fold such that they can bind a small antigen fragment. If these proteins contain an antigen fragment from an infecting virus, cytotoxic cell will bind to it with their T cell receptor and CD8 receptor complex.